Clonoresis is a Chinese liver fluke, and it's common main parasite. The eggs are yellowish brown color, and the eggs of chlorosis sensis is the smallest egg in whole pathogenic parasites, and these are a small knob projection on the end of the egg, and Mycelium is hatched to the after the entering the snail host. Serratia then entered to the fish by penetrating the skin, and from the metaserratia infection into the man caused by the ingestion of raw or uncooked fish in metaserratia. It's mostly caused the colonogingitis, fever, chill, jaundice, pancreatitis, and detection is characteristic egg in the feces and aspirated bile sample. If you see the knob, which is clear here. Trematode are further some flukes, and that is most common are Paragomenius westermanni. It's usually ingestion under cooked crabs or crayfishes, and location of the adult worm is lungs. The ova in the feces are the sputum, is yellow brown, thick shell, flattened operculum, and Chinese wise. And Cystosoma mansoni is through the skin penetration, and intestinal venols, and ova is in the feces elongated and pronounced lateral spine, and it have a large lateral spine, and it is a blood fluke, Cystosoma mansoni, and it's just like a flask, a Chinese flask. You can find it here. Cystosoma japonicum is also through the skin penetration, and intestinal venols are the location of adult worm, and just like the Cystosoma mansoni, and the ova is in the feces and minute lateral spine. If you see the small lateral spine, difficult to see, and developing metacidium is inside the egg. It's also the blood fluke, and the only difference between the mansoni and japonicum is the large and small lateral spine. The japonicum has a small lateral spine, as you see, which is difficult to see, and mansoni have a large lateral spine. Another trematode are Cystosoma hemabotium. It's the root of infection is skin penetration, but the location of the adult worm is bladder venols, not intestine, which make it different, because this organism can only be detected in the urine, not in the stool. The ova is in the urine, that is terminal spine. Terminal spine, if you see, it's a large terminal spine which differentiated from the both species. It is also blood flukes, only parasite of the urinary system. So the development metacidium is inside the ova, which is clear. Paragomenius westermanni is a charcoal leaden crystals are found in these trematodes, and it's mostly found in the crab eating population, as we explained previously, that the first life cycle in the three different species. Number one is snail, which is water egg enter into the snail sporocyte, and redia and serratia formation with the knob-like tail, and then this crabs or crayfishes, serratia enter into the crabs and insist into the mussels, crabs, and crayfishes wide bar host, peritonic host. You can see it is perotonic host. And then in the main, the adult worm in the human lungs are thick reddish brown, and the human infection takes place by ingestion of these raw uncooked crayfish and scrubs and well bore meats, and the parasite exist in the duodenum and penetrate into the intestinal wall and proceed to the lungs, and by penetrating the diaphragm it causes a symptom like the pulmonary TB, endemic hematopsis, and also cause the ectopic manifestation diagnosis is made by the detecting characteristic egg in the sputum and bronchial wash or aspirate pleural fluid are in the feces. For the ectopic infection, serological method is useful. Paragomonius Miyazaki is resembled to the Paragomonius westermani but more elongated than it, and diagnosis is also similar to the Paragomonius westermani. Cystosoma species is, as you know, that is Cystosoma japonicum, hematobium, and mansoni are the species of medical importance, and the female worms are thinner and longer than the male, and all above species are differentiated on the basis of their size and Cystosoma completed life cycle in the human and snail host. The eggs in the water into free swimming meridium to the specific snail host, and finally give rise to the infective Cystosomali. And the human infection takes place by the skin penetration by the Cystosomali while swimming, irritating the rice field, or washing clothes, or ebullition. And this cause the abdominal pain, diarrhea. Mansoni, if you see, this is the picture of Mansoni. It is clear previously. Japonicum, and you can see the Mansoni and Japonicum are the blood flukes, which is mucus in the stool specimen. Hemabotium is a hematouria, cystitis, and burning sensation, while urination of this infection with the Japonicum is most grave. Japonicum is most infective. Diagnosis is made by detecting characteristic in the urine, hemabotium, and the stool specimen Mansoni and Jicum. Also, immunological methods are available. Blood and tissue protozoans are very important in diagnostic parasitology. Acant amoeba, it's amoeba in the swimming into contaminated water, inadequate disinfectant, contact lenses, and the stain smear of the culture material, histological examination of the brain and troph and cyst in the coronal scraping. Causes the granulomatous amoebic encephalitis and ulcerative acant amoeba keratitis in the contact lens wearers, usually in immunocompromised or debilitated persons. And Naegleria fluori is also an amoeba. It's found in the swimming in contaminated ponds and streams, and stain smear of the culture material troph in CSF, causing the primary amoebic meningoencephalitis. And Plasmodium is also a sporozoan. It's not amoeba. And Anopheles mosquito is the transmission route, and right stain, thick and thin blood smear draw the blood just before the paroxysms, causes malaria. It's a parasite of RBCs. Babesia is also a sporozoan, and its transmission is through the tick bite, blood transfusion, and right stain, thick and thin, just like a malaria is used, and the symptoms are resembles to the malaria. If you see the parasite in RBC, it's the ring form, and tetrad or Maltese cross form is also observed in this case, of which differentiated from the malaria. Toxoplasma gonodi is a coccidian, and it is also blood and tissue protozoan, and ingestion of undercooked meat or oocyte from the cat feces, transplacental. It's transplacental and organ transplant, and serological test and PCR is the only diagnosis for the Toxoplasma gonodi. The cat is a definitive host. Congenital infection causes birth defect and mental retardation, most major of encephalitis in HIV patients. And another organism is Leishmania, which is flagellates sandflies, the transmission route, and amostigote form in the WBC and cell of the reticular endothelial system. Its obligate intracellular parasite cause the Kala-Azar, cutaneous and mucocutaneous leishmaniasis. If you see the picture, this is the picture of the WBC, in which the macrophages and amostigote form and macrophage nucleus, and inside that intracellular Toxoplasma gonodi organism is present. In blood and tissue protozoan, trypanosoma is also very important organism. This is also flagellate, and the transmission is through the testifly, which is T uh, Gambinese and rhodinensis, and T. cruzi, riveted bug or kissing bug. The T. gambensis or rhodinensis are treptomastigote in the blood and lymph node and CSF. T. cruzi is a treptomastigote in the blood, amastigote in reticular endothelial cells, myocardium, CNS. The other are T. gambicis and rhodinensis is sleeping sickness, while the T. cruzi cause the Chagas disease found in the southern US, United States. If you see the trypanosoma uh, sleeping sickness, which is caused by gambis or rodents, which is streptomastigote, or in this case A mastigote form is not present, while in the T. cruzi Chagas disease the A mastigote form is present, which is in reticular endothelial system, myocardium, and central nervous system, and that causes the sleeping sickness. That's why it's unable to cause the sleeping sickness, because the amastigote form is in reticular endothelial system, myocardium, and central nervous system, and tryptomastigote form are not there. It's mostly in the blood. So in the case of T. gamensis and rhodinensis, which cause the sleeping sickness, the tryptomastigote form is in the blood, lymph node, CSF, and that's why it caused the sleeping sickness or Chagas disease. If we see the picture of trypanosoma, you will find that the large terminal kinetoplast and also undulating bodies, which differentiate it from the leishmania. It's a unique property of trypanosoma species only. And now we will explain how to differentiate the Plasmodium. Plasmodium falciparum, malaria, and vivax, and you can see the name of the disease is malignant malaria in the case of falciparum. In the case of malaria, it's Cotoran malaria, and the case of oval, it's oval malaria, and vivax, it's Triton malaria. And the peroxisome cycle is in falciparum is 36 to 48 hours, while malaria is 72 and oval is 48, and vivax is 48 to 44 to 48 hours. And RBCs are infected in all of the cases, but the shape of the RBCs are not changed in the falciparum and malaria, while vivax and will change the shape of RBCs. RBCs are enlarged and sometimes frequently will with ragged margins, Schaffner dots is also there in the oval and vivax. And the stages are ring form and trophocyte and gametocytes in the falciparum, while schizont form is not there in falciparum. In malaria, oval, and vivax, all forms are available. Merocyte per mature schizont not seen in the peripheral blood film in the falciparum, while in malaria, oval, and vivax 6 to 12 days, and after 18 to 12 and 12 to 24 days. In the RBC you can find the merocyte. You can find it here. Is the picture of merocyte. Further, the shape of the gametocyte of the falciparum are crescent. You can say the banana shape, and the malaria have ovoid, and oval have round, and vivax have round. The only difference between the oval, the only difference is a falciparum, and malaria have oval, and both oval and vivax are round gametocytes. P. falciparum have quite similar property, just like a babesia, which have multiple parasite purcells, and while malaria, oval, and don't have this property. Vivax rarely show this property. A double chromated dots are only found in the falciform, and it's very rare in the Vivax, while oval in malaria doesn't show this characteristic. And other characteristic of falciform are highly mortality and medical emergency, while malaria is rarely fetal and band-shaped trophocytes, while oval have less common, rarely fetal, may cause relapses, and the Vivax is most common, rarely fetal, may cause the relapses. The only high mortality is falciform. Some other blood and tissue elements, the most important organism are Toxocara, which is nematode of the dog and cats, and transmission of the eggs from the dog and the cat feces, and serological test is available for diagnosis. Human are accidental host larvae in the tissues, and visceral or ocular larva margin which is called toxocarasis. And Trinella spiralis is a nematode, and undercooked pork, meat, or warless or beers, and its larva encysted in the muscle, and serological tests available for diagnosis is xenophilia, edema of upper eyelid, and fever, malaria. Visceria bancrofti is a nematode which is caused by the mosquito. Burgorrhea milli is also a nematode. Transmission is through the mosquito. Microfilaria, both are microfilaria in the blood, and Ginza stain thick and thin smears are diagnostic index for this microfilaria. It causes the encephalitis in the case of Bancrofti, lymphatic filariasis, and in the case of Burgorrhea milli, it causes encephalitis, but nocturnal periodicity, like it from the 10 p.m. to 2 a.m. It's available in the blood circulation. Nocturnal periodicity. Lola lola is a nematode, and mango fly, chrysopsis. It's a microfilaria in the blood. Gimsa stain thick and thin smear, and loosis is nocturnal periodicity, just like a Burgorrhea milli. And another is Onchiria volvulus, is black fly, breed in the fast flow rivers. Rivers, microfilaria from a skin snap or aspirate of nodules, and liver blindness. And that's why just the fly is in the river, breed in the river, that's why it's called liver blindness. Echinococcus granulus is cystoid, ingested of the fecal material from the infected dogs, and radiological ID of the cyst or ID of the scoliosis in the needle biopsies. Material from the cyst, hydrated sand, and serological methods are available. Humans are accidental intermediate host. Cysts develop into the liver and the lungs. Blood and tissue parasites are protozoans, and you can see the Plasmodium species P complexa. It's a Plasmodium malariae falciform Vivax, and the host, intermediate host are man. Definitive host is Anopheles mosquito, and number one is sporozoites, which is ingested by the female Anopheles mosquito, and liver schizogony, and pre-erythrocytic schizogony, and merozoite release from the liver schizont, and then erythrocytic schizogony, and most important on diagnostic point of view. And then the ring form, trophozoite form, schizont form, gametocyte, all inside the RBCs, oocyte, gametocyte, male and female, fertilized to form the oocyte, and ingestion by the mosquito and undergo the oocyte, and infection again circulate. In this case, the Vivax and oval, some of the sporozoite reach the liver cell, remain in the hypozoite for responsible for a relapsing latency period. P. Vivax and oval, and generally schizont of the P. falciform is not seen in the peripheral blood film. It is gametocytes are typical banana shape, and P. Vivax and oval generally infect the young RBCs and are bigger in size, while malaria and falciform have different. Malaria infect the mature RBC, and falciform infect both mature young RBCs in the circulation. Malaria also transmitted through the blood transfusion and transplacental congenital malaria. On a molecular level, the parasite damage the red blood cells using the plasmipsine enzyme and aspartic acid proteases, which degrade the hemoglobin. P. inolesi is known as the severe quotidian malaria in the Southeast Asia since 1965. Merozytes have a schizoid ring appearance due to the large vacuole that force the parasite nucleus to one pole, and schizoids are around to inclusion that contain the deeply staining merozytes. Toxoplasma gonorrhoeae is also epicomplexa. Malaria is also epicomplex, and Toxoplasma gonorrhoeae is cosmopolitan and affected worldwide range of animal, birds, mammals, feline, and cat and dogs. Males are definitive host, while all other animals are intermediate host. The feces of the definitive host oocyte contain the 8 sporocytes enclosed within two sporocytes, and each contain the 4 spore ingested by intermediate host. Phagocytic cells multiplication and pseudocystic crescent shape viridocyte, which is slowly proliferating form, and latent form, and trichocyte, which is rapidly proliferated form, enclosed into the cyst found in the tissue including the brain, and one oocyte are 100 trichocytes, and this slowly enlarged cyst are enclosed into the tissue including the brain, and oocytes containing the dendrobarocytes. It's caused the latency period, that's why not responding to anti-parasitic agents. Also cause the placental barrier, causing the pregnancy infect the fetus and enter into the ingestion of definitive host enterocyte, undergo the merogeny and gametogeny, finally produce the oocyte, pass in the feces. These trichocyte and peridocytes are infective, both definitive and intermediate host.